Okay, let's jump straight in. We got some really cutting-edge science to talk about today from a 2025 paper in the International Journal of Nanomedicine.
Yeah, this one looks at a fundamental problem, getting things like DNA vaccines into our cells effectively, and just as importantly, safely.
Exactly. Delivering that genetic instruction manual, getting it past the cell's defenses, that's the tricky part. So our mission here is to unpack how this study tackles it, using, believe it or not, tiny gold particles.
Gold nanoparticles as a delivery vehicle for DNA vaccines. We need to understand how it works, why it's potentially innovative, and maybe what it means for things like, well, the study focuses on norovirus.
Right. So let's break it down. The main characters here are, first, the DNA vaccines themselves, bits of genetic code teaching your body to fight something off.
Then there's this polymer, polyethylenimine, PEI for short. It's often used because it interacts well with DNA in cells.
And the stars of this particular show, gold nanoparticles. Tiny, tiny bits of gold.
So the researchers didn't just use them separately. They cleverly combined them, creating PEI conjugated gold nanoparticles. Let's call them AuPEI-NPs.
Okay, AuPEI-NPs. Basically, gold particles dressed up with PEI to carry the DNA vaccine.
And there's a neat detail about how they even made the initial gold nanoparticles. They used a green synthesis method.
Green synthesis, so like more environmentally friendly.
Yeah, essentially. Using citrate ions. It tends to be simpler and avoids some of the harsher chemicals you might find in other methods. Citrate helps control how these tiny gold structures form.
Okay, greener gold spheres, then they have the PEI. Why PEI? What's the magic there?
Well, think about charges. DNA has a negative charge. Cell membranes, also negatively charged.
Ah, so like repelling magnets, negative pushes away negative.
Precisely. Getting that DNA payload past the cell membrane is tough, but PEI, it carries a positive charge.
I see. So coating the gold nanoparticle with PEI gives the whole thing a positive charge.
Exactly. And that positive surface charge is much more attracted to the negatively charged cell surface. It dramatically increases the chances the cell will just sort of engulf the particle.
Enhanced cellular uptake, as they call it, making it easier for the cell to eat the nanoparticle carrying the vaccine.
It's tuning the surface chemistry, and the results they reported, well, they're pretty striking.
Yeah, tell us about those. In the lab tests, in vitro, what did they find?
Okay, so in vitro, in cells, in a dish, they hit 100% transfection efficiency.
100%, meaning the DNA got into all the target cells. That sounds almost too good to be true.
It's exceptionally high for this kind of work. But here's the other absolute critical finding: zero cytotoxicity.
Zero toxicity. So it got the payload in perfectly without harming the cells themselves.
Yes, that's often the huge trade-off with delivery systems' effectiveness versus safety. These AuPEI-NPs seem to manage both, at least in these tests.
Okay, zero toxicity is massive for safety. Did they test it in living systems too? In vivo?
They did. They moved to mice. And in vivo, they saw strong immune responses generated by the vaccine delivered this way.
What were they targeting specifically?
Human norovirus, HuNoV, especially the GII.4 variants, which are, notorious for causing widespread outbreaks and evolving rapidly.
Right, norovirus is a tough one. So what kind of immune response did the mice show?
They saw significant NoV-specific immune responses. That includes antibodies, specifically IgG antibodies, and also T-cell responses.
IgG and T-cells. Remind us why those are important against a virus like norovirus.
Well, IgG antibodies are key players in neutralizing the virus particles directly, kind of tagging them for destruction. And T-cells help coordinate the immune attack and kill off any cells that do get infected. It's that two-pronged defense you want.
Got it. So for you listening, the big takeaway here seems to be a really promising delivery system. Biocompatible, meaning it works with the body, and non-toxic.
Exactly. It opens doors not just for DNA vaccines, but potentially other gene therapies where safe, efficient delivery is paramount, especially for tricky targets like rapidly changing viruses.
And the study even hints at broader uses, right? Like targeted drug delivery in general.
Getting any kind of therapeutic agent precisely where it needs to go, potentially minimizing side effects. It's a platform technology, really.
So to sum up, this research highlights these AuPEI-NPs as a highly efficient, non-toxic way to deliver DNA vaccines, showing real promise against norovirus in their tests.
A potential step forward in nanoparticle delivery systems.
Which leaves us with a thought. We know how fast viruses like norovirus change, and we always need safer ways to deliver therapies. Could non-toxic, efficient systems like these gold nanoparticles fundamentally shift how we tackle not just vaccines, but maybe a whole range of treatments needing precise delivery inside the body. Something to keep an eye on.